It's good. So I think it's an auspicious day to start the first day session on Introduction to Bioinformatics and Computational Biology. Dear participants, as Professor Praveen Ramanujam sir has mentioned, try to click yes, okay, in the invitation sent to your email ID. And just try to log in only the email ID what you have given for registration so that it can avoid time, okay, because we have a lot of concepts to be covered in online internship and certificate course in bioinformatics. So if you start from the first day to the 14th day, okay, we are going to bombard with so many concepts unless you are familiar or you have some kind of awareness on bioinformatics, you can able to cope up. But still, I can make myself to relaxly make you understand. And if you have any other doubts, okay, you can message me, you can mail me, you can contact me at any time. Okay, so before starting on this session, that is the first session on bioinformatics and computational biology, where I basically take you on the fundamentals. Okay, and then I start taking on the next 2 days on biological databases, the types of databases and the types of formats and the recent databases what we are using at present for research. And then we go on sequence analysis for 3 days. Okay, so that is we are going to see what are the types of sequence alignment and what are the uh, amino acid substitution scoring matrices, and we see some algorithms because bioinformatics basically is about algorithms. So one should know how the algorithms are developed. Even though if you feed some kind of an input in the software tools and you get some kind of an output, but one should know how the algorithms are virtually going to work to produce a kind of an output if you feed an input. Okay, so that we see some algorithms examples during the sequence analysis, and we discuss on molecular phylogeny, that is how to construct the phylogenetic trees, okay, based on the evolutionary relationship and what are the different methods used. Let us see one method in detail, like how the uh, tree is constructed based on stepwise methods. And then we go across the macromolecular structural analysis, that is on protein structure prediction, visualization, validation of the protein structure predicted, and how to assess the stereochemical quality of a protein. So all these things we'll be seeing on the 2 days. And the last 4 days we discuss on various applications of bioinformatics and computational biology. Okay, so this is how you can find the course contents. Okay, so generally, if you see bioinformatics, okay, we have so many courses from life sciences also, like BSc bioinformatics, MSc bioinformatics, PhD bioinformatics. Okay, so we have plenty, isn't it? And similarly, even in engineering, we have in the form of BTech, MTech, etc. So we have that much concepts. It's like a ocean. Okay, we are dwelling in the ocean, and if I want to make you understand, I have to teach you the fundamentals. So whatever I'm teaching you is the fundamentals. Okay, so these are the fundamentals which I'm going to take you for the 14 sessions. Okay, and if you come across the chapter one, the chapter one is basically on Introduction to Bioinformatics and Computational Biology. If you see what are the certificate courses we have conducted in collaboration with Institute of Innovations, Tiruvannamalai. So these are some of the courses I have shown. So one such course I also have conducted in SVC in collaboration with IIT Madras as well as with software companies. Okay, and we have conducted totally up to present nearly 10 courses we have conducted. And these are the latest courses conducted in the last 4 months. Okay, I mean last 6 months. That is in 16th to 30th May 2024. They have conducted certificate course on artificial intelligence and machine learning approaches for drug and vaccine design in cancer. And we have also conducted 1 month internship and certificate course. Like, however, I just am going to taking a 15 days course. This we have taken for a 1 month, so including giving an hands-on experience on how to handle the projects, bioinformatics projects. Okay, and so whatever earlier certificate courses handled in collaboration with SVC and the Institute of Innovation Tirumalai, we can have nearly 10 courses or 10. Okay, and then if you come across the sponsored workshops and seminars in this decade, in my institution, see SVC, Venkateshwara College of Engineering. Okay, in 2022, I think we have received CS, we have received sponsorship from CSIR to conduct a workshop related to genomics data science in precision medicine design. and an online certificate course in computer drug design, which I've conducted in collaboration with a software company. And uh, this is a popular lectures in biotechnology sponsored by DBT to conduct on anti-cancer drug discovery. So almost all the courses, whatever I take, is related to bioinformatics specialization. And the last year, December 2023, I received a grant of 3.5 lakhs to conduct ASD sponsored Atal FTP. Okay, so during this time, so we got an excellent sponsorship to conduct on this theme, unraveling molecular mechanisms and artificial approaches of drug design in cancer. Okay, and then so related to the sponsored projects and research consultancies for past 3 years, so many projects we have received in the past 3 years. Okay, we got even a kind of a consultancy charges from different colleges like Shri Krishna Arts and Science, a Tech, a PhD scholar has going to complete her PhD under my guidance. Even though I'm not her guide, I'm just guiding her through the consultancies. So I'm guiding a kind of a computational project related to immunoinformatics. And we have received even grants from Tamil Nadu government. So this is the maximum fund they provide for a UG student. And this is a DST funded project which I am executing on and it's in live. Okay, so in this project, I'm basically using a computational skills. That is, if you see this TST funded project, I am linking with IIT Madras to work on computational skills. Okay, so I'm working in protein bioinformatics lab under the mentorship of Professor Michael Komiha, who is India's number one bioinformatician. He has developed numerous databases and tools in his lab. So I'm just working under his mentorship. And this is the recent fellowship received by my PhD scholar last month. So she is going to receive around 6.72 lakhs. Okay, so each and every month she will be receiving 18 to 20,000. Okay, based on the performance. And also recently I received a grant of 5 lakhs. So for 24 to 25, I received from AICT. So even my PhD scholar is going to link with me to start a startup company to market the nano trucks. Okay, so for all the in all these projects, I have utilized the computational skills. So why I'm showing you all this, because we have a lot of opportunities, especially in anti-cancer drug discovery. Okay, if so, how to make a kind of a fruitful solutions for the present problems. Okay, and if you see some of the other achievements or from Truck Discovery Hackathon, okay, which conducted in 2000, and these students from when they are studying second year BTech biotechnology, when they don't have any knowledge on biotechnology. So I just started from the basics during the pandemic times, and I started what is biotechnology, what is cell biology, what is pharmaceutical, what is immunology, what is bioinformatics. They learned everything in a span of 3 to 4 months. With rigorous training, actually, they won the phase one event. So they secured 3.6 lakhs. Each and every student has won nearly 60,000 as an internship stipend. So total worth 3.6 lakhs. And if you see the another achievement in 2022, so both is funding we received one in 2022. In 2022, we have won Innovation Protein Hackathon. My PhD scholar has secured 1.07 lakhs. So she whatever she secured, okay, that 1.07 lakhs. She also won the first prize out of the 20 prize winners selected from Good Food Institute, India. So she secured the top scorer, and of course, she also won the prize amount. And many students, UG students, have secured the first prizes, etc., in the computational competitions. So about the recent event, what we secured, and presently I'm working this live project. This project is in live. So this is on a Vithnot A, potent inhibitor of mitochondrial HSP90 of hepatocellular carcinoma. So actually here I'm using the computational technique, and of course I'm using a nanotechnology delivery. So here if you see recently from the IIC, Institution Innovation Council, so we have received nearly 1,05,000 submissions, only 135 innovation secured grant. So my innovation is out of is one of the 135 innovations. So we have a lot of opportunities, okay, if you succeed with your skills. Now let us Before starting with this computational biology and bioinformatics basics, I just want to get a kind of information, okay, from you through polling. Like, I want to know how many or having a UG background, how many have PG background, and how many have PhD background. Okay, so can we start polling at present? So for 1 or 2 minutes, okay, I just ask a question like, so kindly provide me your education background because I'm going to teach you within a span of 14 hours, that is 14 days. I don't know how you're going to, how you can able to understand, how much you can able to understand. Okay, I want to check. Okay, so the polling is started. So see, students, actually a good practice is to accept the invitation because I like to teach you much concepts, and if I just started the poll, isn't it? Can you able to see this polling? Yes. So UG 33, everyone can try to vote. I want to check the numbers. So we can see the ratio for the 61 votes. Still, we have 105 votes. I have to receive 105 votes. Are you listening to my lecture? If you are listening to my lecture, interact with me. So this is most important, participants. Okay. Even many senior professors are also attending. I know the dean of from a popular university. She is also taking. She has also received many awards, national and international awards. She also would like to join this course because the real learner, the enthusiastic learner, can learn at any ages. We won't restrict. Okay, just because you can't poll, I think you have mentioned your qualification. Good, madam. May know, madam is okay. And how about the others? So we can able to find here. Okay, if I add 32 plus 11, around 43. So we have advanced learners. When compared to UG, the PG and PhD combination is more, isn't it? So I can take you, but if you don't understand, okay, you can question me. Okay, you can raise your hands so I can stop, or you can better drop whatever you don't understand in the form of a comment in the chat box, so I can explain it. Okay, so even I can accumulate it and I can cumulatively explain with all the answers at the end. Okay, where we see poll box. Actually, the poll box you can see in this triangle, square, and circle after the chat box. After the chat box, okay, you can find this activities. In the activities, one can able to find the poll box. Okay, the PG and PhD are dominating, however. So I got this. Um, can I end the poll? Because I have to come across. I got it. Still, the PG and PhD are dominating. So end the poll. Thank you for your interactions. So next time, even you can alert. Okay, so if I can interacting with few questions, try to use this kind of an option. Okay. Now let me start with the lecture. So I was explaining you something about the computational biology and bioinformatics. Actually, our course is on bioinformatics. But before going to this, okay, if you want to connect with me, okay, I can also show you my profile. So this is my profile, LinkedIn profile. I'm sharing it to you. If you want to connect with me, you are welcome. Okay. So I just sharing you the LinkedIn. Okay. Now I just start taking the class. So bioinformatics, there is also one more term called computational biology. What is the difference between these two terms? So if you start taking about the bioinformatics, many of the universities offer subject like computational biology and bioinformatics. Okay. If so, I think I'm getting some kind of chat information. No. Okay. So if you're getting something like computational biology and bioinformatics, the computational biology basically we involve the biology. Okay. I mean, we use the computational studies to study the biology. What are the computational techniques? That is, if we have created a software, okay, to interact with a data sets, okay, to study about specific types of biology, can be cell biology, immunology, okay, molecular biology. If you use a computational tools to study these types of biology, we call that as computational biology. Then what is bioinformatics? The bioinformatics is not just the application, but it is a creation. It is a creation of tools along with algorithms and databases. Like whatever databases you want to create, or if you want to create tools like Blast, FASTA to work on sequence alignment. Okay, and if you want to solve problems, okay, obviously one has to choose the bioinformatics. You got it? Again, in simple terms, computational biology connotes the development of algorithms, mathematical models, and methods for statistical inference. But whereas bioinformatics is more associated with the development of software tools, databases, and visualization methods. Now, let us come to the some areas of informatics. One is chemoinformatics. What is chemoinformatics? It combines the scientific working fields of chemistry, computer science, and information science. Here you find chemistry, computer science, and information science. Okay, that's what we call it as key informatics. Okay, so we are applying all these things you apply in the areas of topology, chemical graph theory, information retrieval, and data mining in the chemical space. So more about this, if you see in the subsequent slides, you can understand. And like how I just split and explain all this informatics. Similarly, if you see the bioinformatics, bio, default we take molecular biology, info, information technology. So in information technology, we basically use bit information theory, isn't it? Whatever data we store in the form of bits. That's what bit information theory. So info refers to the information technology, and matics here basically we use statistics. If you have plenty of data, especially if you see the big data, the big data in the world, if you see which area is covering the most of the big data, you find bioinformatics is the area where you can find more big data. If so, to handle all these data, one should have an information on information technology, that is bit information theory. Okay, so you got it. So bioinformatics comes from three areas of science. One is biology, molecular biology, information technology, and statistics. Likewise, if you see the cheminformatics, other than the molecular biology, better use chemistry. Okay, we study more on chemistry. That is on chemical compounds chemistry. We go with chem informatics. Okay, we all majorly use this chemoinformatics in the pharmaceutical applications. So more about this we see in the subsequent slides. Basically, for drug design, one can be able to use the chemoinformatics. For example, in this slide, you can able to see a database called NCBA PubChem. In NCBA PubChem, if you want to search for any drugs for a specific disease like COVID-19, so COVID-19 is considered as one of the keywords. Okay, like if you type COVID-19, obviously can find 1,581 compounds. At present, I think they have increased. I think this extract is taken some 1 or 2 years before. Now, I think around 1,700 or 1,800 compounds are increased. What are these compounds? These are the compounds generally used for treatment of COVID-19. Okay, so whatever compounds you can able to find here after typing the keyword like a disease name, you can download all these compounds in the form of a single data set. So whatever data set you download, okay, you take all the compounds in a single file in a single data set, and you can go for visual screening of compounds to find which is best for the particular disease. Even if you take a particular disease, that particular disease is not just caused by a single target receptor. Okay, there are various protein targets responsible. Okay, one can go to the protein target specifically, and then you can screen the compounds based on the bind. So, however, whatever I explaining, like compounds, taking from from a keyword, like typing a disease name, okay, or even you can type a compound name here in the search. Okay, if the any specific compound, okay, if any turmeric compounds, neem compounds are also treating the COVID-19, type the compound name, okay, and then you just find the structures of the compound also. Okay, so this is a largest database for a collection of compounds. So that is, you can find the chemistry of the compounds, and you can find even the physical and chemical properties of the compounds. So, like NCBI PubChem, we have few more chem informatics databases like Chem Spider. Okay, the Chem Spider, you can type the compound name and you can find the detailed annotation of the chemical compounds. Like if you want to work on drugs, okay, existing drugs, repurposing drugs. Initially, before coming with a drug for SARS-CoV-2, many of them tried with the repurposing drugs. If you want to know about the repurposing drugs, one can go to the drug bank. This is the largest database for the drugs. Okay, why we are using drug bank even though we have PubChem? So in drug bank is a more annotated database. That is, you get more information when compared to PubChem. Like you get almost all these details like pharmacology, interactions, products, categories, chemical identifiers, clinical trials. So one can explore in more, okay, using the drug bank. Of course, even NCBI PubChem, it's not a small database when you can find much information. This is especially meant for the drugs. And if you see the next database is CHEVI. Okay, this is from European Bioinformatics Institute, also mentioning a chemical database. Okay, these are all chem informatics databases. So whatever compounds you have taken from NCBI PubChem, if you want to check the drug likeness, okay, so to check the drug likeness, we can able to do with chem informatics tools. So till now, whatever I showed, like EBI, CHEBI, NCBI PubChem, Drug Bank, all these are chemoinformatics databases. So this is a chemoinformatics tool. What is that? MolSoft, Mol Inspiration, Swiss ADME, etc. This is going to justify whether your compound is behaving like a drug or not. Compound, drug. What is the difference between compound and drug? Compound, basically, here I mean can be taken from sources like plants, or if you have taken compound from the algae, if you are working on algae, and if you want to check whether that algae compound is having a drug likeness property, so one can able to first get the properties, and then you can check with this drug likeness filter. The drug likeness filter, what I've shown at present, is the Lipinski rule of 5, also known as Pfizer's rule of 5. Okay, so according to this rule, we have four important rules. What are the four important rules in the Lipinski rule of 5? The first rule is molecular weight. That should be less than 500 gram per mole. The second rule is log partition coefficient. That should be less than 5. The third is hydrogen bond acceptor. That should be less than 10. And hydrogen bond donor less than 5. These four alone, we call it as Lipinski rule of 5. Then why we call it as a rule of 5? Because all these four properties are present in the multiples of 5. Just because it's available in multiple of 5, we call this as Lipinski rule of 5. Okay. So if a compound has to behave like a drug, it has to satisfy any of these three properties out of four properties. If a compound is satisfying this property and this property and this property alone, still we can tell a compound behaves like a drug. But so even if it follows this, there is no doubt, of course, the compound is well behaving like a drug. Okay, because some compounds are not desired. So if you take a plant, neem plant, the neem plant, everyone know it is a base, it's having a best antiviral properties. You may have hundreds to thousands of compounds in neem. But if you take an antiviral compounds, you may have 100 compounds, or around 100 compounds, and some other 10 or 15 compounds or not giving an antiviral property, but it's going to block the antiviral property. If so, instead of taking all the compounds together, we can just isolate the antiviral compounds alone to get rid of the other compounds which is not involved in antiviral activity. You got it? If you are checking for an antiviral activity, especially for SARS-CoV-2, that is for a particular strain of SARS-CoV-2, one has to consider the antiviral property. Okay. So if a compound behaves like a drug, it has to satisfy any of these three rules. And what is this, the other thing? This is rotational bond less than 10, and polar surface area less than 140 angstrom. So these are the other rules which are present in other drug likeness filter. Now, presently, whatever I explained is about one drug likeness filter called Lipinski rule of 5. So in other filters like Faber, Moose, we can also find the involvement of these physical properties of drugs. I mean, chemical properties of the drug. This is a physical property linked with a chemical property. This is a chemical property of it. Got it. So these are some of the properties we consider for drug likeness prediction. And of course, this is a best example for key informatics tool. One such output from drug likeness predicted from MoleSoft. Okay, you can find hydrogen bond acceptor uh, 6, which is which is lesser than 10. So this is acceptable. Hydrogen bond donor should be less than 5. So this is also acceptable. Molecular weight should be less than 500. So this is also acceptable. And we have the surface area. The surface area, I mean partition coefficient less than 5. Okay, this is also acceptable. So you can satisfy all these four properties. So you can tell the compound behaves like a drug. You know what is the name of this drug? Curcumin. Curcumin. Okay, so for a curcumin compound is showing the drug likeness property, so we can consider this compound as a drug. And of course, you find a drug likeness score as minus 0.82 from the mole sort. So if you find the values in between -2 to +2, you can tell it behaves like a drug. Our compound is lying in minus 0.82. So just because it is in this range, so this is following a drug likeness property according to mole sort. This is another software tool called Mole Inspiration to predict the drug likeness properties. And I also told you there is a software tool called Swiss ADME, which can predict the absorption, distribution, metabolism, and excretion characteristics of a drug. So this we call it as pharmacokinetics. So whatever you study in pharmacokinetics, if you want to determine using the online tools, so this is why best cheminformatics tool available in the internet. One can go to the Swiss ADME. One can go for pre-ADMET also. There is a tool called pre-ADMET. Okay, to find the chemical and physical properties of the drug, especially pharmacokinetic properties of the drug. Not only find the physicochemical properties like drug likeness properties, but also we can find the pharmacokinetic properties of the drug. Okay, so this is something about ADMET. So output from a Swiss ADME. So as I mentioned, in Swiss ADME, if you just drop the structure or you drop the SMILES format, canonical SMILES format of a compound. In our case, I just dropped a curcumin compound SMILES format. The SMILES, canonical SMILES, is a 1D format of a compound. So that 1D format of a compound, if you drop and if you click run, obviously get a kind of a 2D structure and you get the properties like the molecular weight, which is less than 500, so it is satisfying. And how about rotable bond should be less than 8 or less than 5, but it is showing 8, so it's not satisfying. But hydrogen bond acceptor should be less than 10, so it is satisfying. And even hydrogen bond donor should be less than 5, it is also satisfying. That's because it is satisfying 3 rules out of 4. I can't consider it as rotable bonds is less than 10, isn't it? It's less than 10, not less than 5, it's less than 10. So, so only we are getting 0 violation. The rotational bond should be is also satisfying. That's because all these four properties are satisfying. If you are getting like 0 violation, so only Lipinski, you are getting like S. And we also have other filters like Goose, Weber, and again, here also there are no violations and it shows the drug likeness properties. Other than the physical chemical properties, we have absorption. For absorption in ADME, okay, in absorption in ADME, okay, gastrointestinal absorption should be high. Generally, what do you mean by that? So whatever drug we take, which we consume in oral route, the preferred route by many of the patients is to take in oral route by swallowing the drug, not by taking in the form of injection. So if your drug can be taken in oral route to get an antiviral activity, okay, in our case, so GI absorption should be high. Yes, it's giving a desired property. And here you have the next. This is related to distribution. So the distribution has to distribute to the desired tissues. It should not distribute to the brain. You know what is BBB? Blood brain barrier. Blood brain barrier. Why we are using blood and brain? Actually, the blood, you have pH of 7.4. Okay, and in brain, so it's not just the pH is concerned. Here you are concerned more on the lipophilicity. Lipophilicity. The blood is less lipophilic when compared to brain. You know, the brain is having multi-membrane. Okay, it's a thick membrane. So only the lipophilic drugs alone can able to permeate inside. So if you take any of the drugs, okay, and these drugs, like even if you take a paracetamol, if you take a single dose of a paracetamol, which is generally used for treatment of fever, okay, it can able to target for 8 hours. The residence time of paracetamol in our body is around 8 hours. But instead of taking a single dose, if you are taking 3 doses a day, and if you take all the 3 doses at a single instant, I mean, at the same instant, if you take 3 doses, what's going to happen? You know, even this paracetamol, just because of high dose, okay, it can target the central nervous system. No, participants kindly take care. So here, I'm just giving a safety procedures. Okay, I'm just teaching you only in positive thing. Okay, don't take anything negative here because even if you take more than 3 doses, or even it can the people can also lose the eyesight. If you take 1 or 2 doses, even it can end their life. So the same drug paracetamol can use for treatment of fever. If you take in excess doses, you may lose vision, you may lose even the life. You got it. So we have to use a preferred dose. If you should not go beyond that. And even if you take a drug which is generally used for treatment of relief from viral infections, instead of uh, attenuating the kind of antiviral activity, okay, if it's going to penetrate the brain, then it's also leading to the central nervous system disorders. So the blood-brain barrier should be no in our case if you are targeting virus. So that is a desired property which comes under distribution. And in metabolism, we generally go with the cytochrome P450 enzymes in liver. You know, the metabolism majorly occurs in the liver. So in the liver, you have so many cytochrome P450 enzymes. And if your drug should not be metabolized much, because if you want to target much of the viruses in the blood, you should not metabolize much of the drug in the liver. So you should get a no kind of a characteristics. Of course, you're getting no, except one. We are getting no. Okay, so this is also desired. Okay, and excretion. The excess drug is going to eliminate in the form of urine or even in the form of a sweat. The skin permeation, you got like minus 6.28 centimeter per second. So these are some of the desired properties what we expect from a compound for treatment of any specific diseases. So this is one more tool called pre-admit to predict the drug likeness prediction, ADME prediction, and you can also, if you want to predict the toxicity of a drug, one can go with toxicity prediction from pre-admit. So till now we have discussed about chemoinformatics databases and chemoinformatics tools, how to use the databases and tools. Okay, now let me take you to the immunoinformatics. What is immunoinformatics? Immunoinformatics, also called as computational immunology, it's a field of science that encompasses high throughput genomic and bioinformatics approaches to immunology. So, if you want to work on immunoinformatics, one should have an essential knowledge on basics of immunology. Then you can able to interpret the results. Usage of immunoinformatics tools and databases is very, very easier. But how to interpret the results deriving from immunoinformatics? That's most important. Many vaccines are developed through immunoinformatics approach, and so whatever vaccines we develop from the immunoinformatics approach is also more precise. That is, if you take SARS-CoV-2, for a SARS-CoV-2, Now I think I can interact with you just because I'm taking you much concepts. The interaction gives you a better kind of understanding. Yes, he's sharing. It's visible to me. Check your network. Okay. So just I'm going to ask you a question. Uh, the question from me is, tell me the vaccines we use for treatment of SARS-CoV-2 infection. So I need answers from your side. Participants can type your answers in the chat box. What are the vaccines? Can Sputnik? Very good. So this is from Russian based, isn't it, Sputnik? Okay, how about the others? What are the vaccines you remember? The government of India has come up with some free vaccines, isn't it? If not Sputnik, the government of India has sponsored us Covishield, Covaxin. So many of them remember these vaccines, isn't it? Covishield, Covaxin. But the next question is, how many remember? Okay, so now this is freezed. Okay, now we can start. Stop. I can ask you the second question. The second question is like, what is the percentage efficacy of Covishield? First, I can ask you about the Covishield for the first strain of SARS-CoV-2. You know, nearly hundreds of strains of SARS-CoV-2 are there at present for alpha, beta, gamma, delta, iota. So many strains are there. For each and every strain, you have nearly 6 to 8 mutations. So whatever vaccine initially discovered for Covishield or Covaxin, or generally that can be used better used only for the first strain of SARS-CoV. It can't be used for the other 99 strains. If that can't be used for the other 99 strains, we have to develop the vaccine. For the vaccine, we have two doses: primary dose and secondary booster dose. After the secondary booster dose, what is the percentage efficacy? The percentage efficacy, the maximum percentage efficacy we found in India is around 82 percentage. But after first dose, it comes around 50 something, 50% is something. But only after the second dose, we find an increase in 82 percentage. If you take the Covaxin efficacy comes around, no, Covaxin comes around 91 percentage. The maximum efficacy what you can find from the Covaxin is around 91 percentage. But still, see, even for the first strain, we didn't get 100 percentage. Okay. But if you take the other strains, if you take the Delta strain and other strains are originated from India, not from the Wuhan, China. We can't blame each and every strain is originated from China. Even some major some strains are also originated, like Delta is the famous strain from India. If so, how whether this Covishield and Covaxin is going to safeguard this Delta? No, we can't able to get any precautionary measures. The efficacy of the Covishield and Covaxin is less than 20 percentage for the Delta strains, less than 20. Then how about the 80 percentage people are not getting any kind of a relief, even though after taking two doses? Then what we have to do? We have to manufacture a vaccine precisely for that strain. One should know what is the structure of spike glycoprotein of Delta strain. Then he has to discover with a vaccine. So why I'm telling you all this, through the immunoinformatics approaches, okay, USA has developed a vaccine called COVAX. One can search this in web, COVAX. This COVAX vaccine is developed through the immunoinformatics approach. So through this immunoinformatics approach, whatever vaccine they have developed, the percentage of efficacy has raised up to 99.9 percentage. So almost all the patients are getting cured. How about the point one is not getting cured? They may have a multiple illness. They may have suffering from heart diseases. They may suffering from diabetes. So if they are facing this kind of specific strains, even you can customize the COVAX. If they are suffering from Delta, instead of a original strain, that is a first strain, so they can also we can also customize the COVAX vaccine to treat the Delta strain. So what I want to mean is, through the immunoinformatics approach, what we call it as a computational immunology approach, we also call this as reverse vaccinology. We use this majorly in a reverse vaccinology approach. So whatever the reverse vaccinology we use to come up with a kind of a peptide-based vaccine is more precise in targeting the viruses and also major types of cancers. Even in cancers, we can use the immunoinformatics approaches. Okay. So this is something related to the immunoinformatics. Majorly, you should have a knowledge on immunology, and then you should have some computational techniques knowledge. So to infer the results, okay, derived from this immunoinformatics approach. So if you say this reverse vaccinology, one should know what is classical vaccinology. So generally, for vaccines, live vaccine, attenuated, killed, whatever type of vaccines we use for the precautionary purposes. Okay, so to get rid of the viruses, bacteria, parasites, so all these things we generally take. If you want to combat the viruses, what we do, we take the viral particles, we take the viral proteins, or we inactivate the virus by imparting more heat to the virus. We use the killed viruses also sometimes to get rid of the active viruses, isn't it? So what we do, we take the virus and we kill the virus. We take the bacterium and kill the bacterium. This is how even the smallpox is get ridden, isn't it? So many viruses or get ridden just because of the introduction of vaccines, and of course, the vaccines main source is you take from the that same viruses itself. Even for SARS-CoV-2, whatever Covishield and Covaxin, what is the major input material we get? We get only the spike glycoprotein or a kind of an RNA, whatever it is. Okay, we take only that for treatment, isn't it? For taking a precautionary measure. Yeah. So, what do you understand? So, however you understand this, this is what a classical vaccinology. We produce it in a bioreactors in large quantities and we supply to the numerous patients. How about in reverse vaccinology? So for getting a vaccine, we are not taking from the virus, but we get a data from different omes. You know, for SARS-CoV-2, a genome is established. Even we have a proteomes for SARS-CoV-2. Whatever you have, a genomes or proteomes. So these are, of course, a data derived from bioinformatics. Okay, so these data, whatever available in the website, in the common public domain, like NCBI or in the Uniprot, etc. We take these kind of a data, bioinformatics data, and from this data, we find the targets, structure, and then we use it for discovery through reverse vaccinology. So one should know the structure information of the diseased protein information. Okay, so to get a knowledge on the development of vaccines. That is, getting a peptide from a protein, disease protein can able to cure, can also attenuate the viruses. Got it? So this is something about reverse vaccinology. That is, we're not taking from majorly from the organisms, but we take from the bioinformatics data. So that's what we call it as reverse vaccinology, designed from information. And this is one such publication I made in 2020. So this is T-cell based epitope based vaccine design for Nipah virus. You know, the Nipah virus infection popularly occurred in India in 2018. I think many of them are aware of this infection in 2018 in Kerala. Okay, even we find a kind of film, a movie called Virus, isn't it? So many problems they face, and even the patients has to isolate in the hills, and just because of there is no treatment, obviously you're succumb to death after a few days, isn't it? It happened. The death is rapid if you get a kind of a Nipah virus infections. But at that time, okay, we don't have much knowledge on that. So a scientist from USA directly came and accidentally he found that it's not accident, incidentally he found the infection from the patient, and obviously many patient lives are saved. And why the Nipah virus has not taken more than 23 or 24 lives? Because the Nipah virus proteins are not getting rapidly mutated. These virus proteins are not getting rapidly mutated like SARS-CoV-2. So, just because of yearly diagnosis, okay, by a doctor called Dr. Ashok, so we are that Nipah virus fatality was drastically reduced during that times. But at the time, I've taken this as an initiative and used a reverse vaccinology approach for a protein called W. So I just worked on protein W in a Nipah virus to come up with a T cell epitope-based vaccine. Okay, so this is an outcome of a TNSST project, and of course selected in a national level award for this project. So this is the sequential workflow I used. One should have a knowledge on protein of what you're going to target to get a kind of a vaccine for Nipah virus. Okay, so I just taken the protein sequence and screened for various T cell epitopes, and the best of I just talked with a kind of a human receptor and come up with which is the best peptide for treatment of Nipah viral infection. And if you see, so this is the end of immunoinformatics. Just given you a basic information of what is immunoinformatics and what is reverse vaccinology. And reverse vaccinology, again, if you want to recap, the reverse vaccinology generally used to discover the peptides, okay, to screen the best peptide for the treatment of infection. I mean, you can able to make it up for a single target receptor. So I just targeted a protein called W. There are plenty of proteins. If you want to target a specific protein, one can also screen a peptide. Okay, make it a different peptide. And whatever peptide you get, that can be best used as a target, okay, for the specific target protein. Okay, and so this is up to the immunoinformatics. Now I'm taking you something related to data life cycle in pharmacogenomics data management. This is an extract from Bioinformatics Computing by Perkiron. So according to that data life cycle, okay. So if you want to create a database, myself, I have created a database in Protein Bioinformatics Lab. Okay. So I've developed a database called Immunoanalytical Database in Sri Venkateshwara College of Engineering. So if you want to develop a database, one should have a different kind of pipelines. Okay, we should have a kind of a methodologies, etc. So here, this is a common concept we can find from a textbook. So according to this extract from a textbook, okay, so here, whatever you find here, okay, that is from here to here, this is actually related to the bioinformatics database management. That is taking a tissue sample, going with the microarray data analysis, and with the pattern and reorganizer, you are going to organize okay, a set of data in the relevant databases, like a gene expression database, medical relevance database, archive, data dictionary. So this we call it as a database, bioinformatics database management. And we also have other database management called clinical database management. So the biotechnology people generally do these kind of an analysis. So instead of microarrays, nowadays we are using RT-PCR analysis. So through the RT-PCR, we are generating so many kind of gene data sets. Okay, so whatever gene data sets we get, we can deposit it in relevant databases. So this is a bioinformatics database management, and the top one which they are showing at here is the clinical database management. So because this in only the clinical database management, one can know about how a drug is going to interact. Like for example, if a person is having an asthma, okay, asthma which is a lung disease, and I am going to give a drug one, okay, the drug one. After giving a drug one, okay, if I want to check the patient status, how well this drug one is going to treat an asthma patient. So one can able to give take a tissue sample and it can go with this bioinformatics data management, and the same patient can be used to get various clinical analysis like taking X-ray, taking other kind of ECG and other kind of lung related analysis. Okay, whatever we want, ECG, EEG, so whatever we want to check, even we can check the other data also. And whatever data is okay, if it is related to ECG, it is related to heart cardiology department. If it is related to blood, okay, may it link to the pathology department. So we can link with the various departments to identify how exactly if the drug is having an effect on treatment. Okay, so this we call it as a clinical database management. So obviously, you take the clinical database management and bioinformatics database management, and you can link with this. We can have a comparative analysis. If and if you comparatively analyze, then you can have an knowledge. And if whatever results you get from the clinical analysis, if it is matching with the bioinformatics, then you can tell the bioinformatics pipeline is more successful. So we can also make a kind of a conclusion based on the comparison of results from clinical and bioinformatics data. Now, of course, if you see the steps in data life cycle, one can able to see the first step as creation, acquisition of data, can be a clinical or bioinformatics data. And whatever we generate the data, one has to use the data. You know, for clinical data in NCBI, we have a database called CleanWare, CILI, CleanWare. This is the database for human clinical samples. And for bioinformatics data, we have data bases like GEO, Gene Expression Omnibus, and then Express Sequence Tags, EST database. So EST GEO databases we have generated from the bioinformatics data, and CleanWare is a clinical database generated. Okay, so we can also use these databases through the proper application suite. Okay, so in NCP we have an application suite. Okay, that is we have an and application suite called Entrus. That is a common interface which we can open all these cleanware or GEO databases, and then we can analyze the information whether the drug is treating successfully or not. And then we can modify. This is the third step, modification. If you want to modify, okay, you can modify the data. And the fourth is to repurpose. Like if you already have one or two drugs, and if you want to check whether this effect of one drug with other two drugs is going to give a best effect or not, one can check that repurposing information through the by giving the repurposing drugs. And the third, whether if you want to repurpose the data, even you can repurpose the data. Okay, so whatever you treat for the asthma, whether it can able to cure the cystic fibrosis, you can also check that. Cystic fibrosis is also a lung disease. So one can also check whether it can be repurposed or not. And finally, the fifth step, you can go for archiving and disposal of the data if data is not in use. So if you want to archive, you can archive. If you want to dispose, if the data is not in use, you dispose the data. Okay, so these are the steps in data life cycle. And of course, this is also an extract from Bioinformatics Computing by Bergeron. According to the Bergeron, okay, we have different types of data models. For example, flat database model we can observe in Notepad. In Notepad, whatever you save the text, okay, we can save however we want. And what we have on the flat file. So the best example is a Notepad. And hierarchical, the best example is XML. You know, XML is a database language where you can store the data in the hierarchical fashion. And if you see this data models, we can use it in various databases like um, object-oriented. That is, so we call the kind of database management we call it as ORDBMS, and relational, RDBMS, detective, and semi-structured. Okay, these are some of the database management models, and more about this we can also see from the textbooks. Okay, so till now I have explained you. Uh, if you want to see the recap, so till now what exactly we have seen, we have discussed on fundamentals of bioinformatics and some areas, fundamentals of computational biology and some areas of bioinformatics like genomics, proteomics. I showed you in reverse vaccinology, isn't it? And then heme informatics. Okay, what is heme informatics? What are the tools and databases present in heme informatics? How about immunoinformatics? and what are the various database technologies. Okay, so this is what we have discussed up to present. Okay, and if you have any doubts, you can ask me at present. I found only one question from Deepanjali Rai. What is log P at drug likeness prediction? Log P is log partition coefficient. The log P generally we use if you want to check in uh, chem informatics terms. Okay, we are using octanol water partition coefficient. You know, chemically also we can able to uh, determine the partition coefficient. That is whether if your drug is lipophilic or hydrophilic. If you want to find your drug is a lipophilic or hydrophilic, you are using a parameter called log P. That is a partition coefficient determination that you are going to determine using this octanol water partition coefficient. You know, the pH of water is so in our body we have blood. Okay, of course, blood is going to have water also. Most of the component is water, but other than water, we also have heme, globin, etc. Isn't it? So if you see the pH of the pH of the blood is equal to the pH of octanol. So if you want to find whether your drug is going to have to find the partition, that is penetration of your drug in the blood, okay, so you can able to find through the octanol water partition coefficient. Importance of the data life cycle. The importance is like if you create a database. So till now, I just showed you like how to go with a database like Chem Informatics databases. You remember I showed you NCBI, PubChem, CHEBI. So these are the databases already established. These are public domain databases. But if you want to create a database, one should understand a data lifecycle in pharmacogenomics database management. That is, if I give a drug, whether the drug is going to treat the patient or not. This you are going to study through bioinformatics database management and clinical database management. Because if you study clinical database management, then what is the use of studying bioinformatics? One should also study the bioinformatics database management. So why we have to study bioinformatics database management? Because already we have data present in the databases. So, studying and analyzing is quicker when compared to clinical. For clinical, one has to newly have or we have to explore how to work with the cleanware databases already present. Okay, so in bioinformatics, you have already existing data. You can work and conclude easily, faster when compared to. So one should also have a kind of a comparison to validate whether your drug is treating or not. Sir, for new virus, how can we work on reverse vaccinology? The data is may not available in the database. Even for SARS-CoV-2, initially many scientists find problem in getting a 3D structure for spike glycoprotein through X-ray crystallography and NMR. But later, the IAC, Indian Institute of Science, Bengaluru, and other top government central institutes have come up with the cryo-electron microscopy. Through the cryo-electron, they came up with the kind of a spike glycoprotein structure within a span of 2 to 3 months. Within a 3 months, we got a kind of a structure. After knowing the SARS-CoV is the major reason for getting the epidemic and followed by pandemic situation. Okay, so you may get a structure, okay, from any of the gold standard techniques within 1 or 2 months. But after getting this structure, one can able to go use the reverse vaccinology to come up with a base peptide for precise targeting. Sir, what about lipophilic nature and hydrophilic nature? So that's what actually in optimal water partition coefficient. Okay, so you can there is one concept called ADME in pharmacokinetics, isn't it? So in pharmacokinetics, what is D? D stands for distribution. If your drug is distributing well to the tissues or not, that you can able to find if your drug is dissolving in the blood or not. If the drug is dissolving in the blood, one has to know what is the optimal water partition coefficient. So we have to determine the optimal partition coefficient. Got it? In starting, some value are given for curcumin only or common for all. It is common. Actually, if you check with a few chemical compounds, you get a a different properties for that. But whatever parameters shown, like the drug likeness filters and absorption, GI absorption, blood-brain permeant, cytochrome P450, these are the common parameters you can get for even the other drugs. Okay. I think I've taken additional 15 minutes on today. Participants from next class onwards, you have to join on time at 7 o'clock. Okay, because even the recorded sessions will be shared by our coordinator and managing director, Dr. Praveen Ramanujam, sir. So you should join the session on time. And my request to all the participants is to accept his invitation in Gmail, whatever you have received. You click yes. If you click yes and if you save the link, okay, whatever link, so this is our link, isn't it? So if you want to join, one has to go to the specific link. Okay, so go to the specific link. Okay, this is the link commonly observed for all the days. Okay, the remaining 13 sessions. So one can use this. Okay, link for joining. Okay, so don't miss this. And if you have any doubts, you can also follow me in LinkedIn or through the mails. Okay, so thank you very much, participants. Have a great day, and I thank you all the participants who have interacted me well with your answers as well as with your questions. Have a great day to you all. Bye.
Thank you, sir.